Um, Hope that you can see me. My name is Carly Ornstein, and I'm the National Director of Lung Cancer Education here at the American Lung Association. I just want to welcome you to our webinar, How to Understand a Lung Cancer Research Study. So today's webinar will be mostly focused on reading lung cancer studies, but even if you don't have an interest in lung cancer, we welcome your participation because the skills you learn today will really help you in reading all kinds of scientific research. So just a bit of housekeeping. I think everyone's done this, but just make sure that you're muted and that your video's off. Both those buttons are at the top of your screen. This webinar will be recorded and it will be available shortly after the conclusion of the presentation. And you'll also see that we've turned on closed captioning for this webinar, and so that's at the bottom of your screen. And then lastly, we're going to be taking questions by chat today. So there's a little chat box that the buttons on the top of the screen, and you can open that up and chat your questions, and we'll get to them at the end. So I'm super excited because I'm joined today by our presenter, Dr. Khadijah Mitchell. And Dr. Mitchell is an assistant professor of biology at Lafayette University. She is passionate about using science to help address health disparities and integrating her laboratory, public health, and teaching work. And I'm so excited that we get to pretend to be some of Dr. Mitchell's students without having to pay college tuition. So it's a real treat. So welcome, Dr. Mitchell. I'll hand it over to you now.
Thank you so much, Carly, for that introduction. So I'm really excited to be here. I'm actually going to share my screen. Here we go. Okay. And so, like Carly mentioned, my name is Khadijah Mitchell. I am the Daubermont Scholar of Health and Life Sciences and an assistant professor of biology at Lafayette College. And I spend a lot of my time designing lung cancer research studies. In particular, I look at health disparities in African American populations. And even though I spend time thinking about implementing and interpreting my results, a big portion of what I do is thinking about how to communicate my science, and not only to my colleagues, but also to the public. So that would be family, friends. And it's really important that as a scientist, we also take time to help individuals understand how to actually read a lung cancer research study and to understand what the implications mean. And so I was really happy that Carly invited me to talk to you all today about how to understand a lung cancer research study. So this is my second time partnering with my friends at the American Lung Association, and I think that you will have a lot of practical tips that you can take with you after today's session. So today's talk briefly has 3 sections. We're going to spend some time at the introduction talking about what are the types of scientific articles. I think we take for granted everyone knows what exactly is a research study and what are the components of that. And then we're going to do a little bit of anatomy. Okay, so talking about a particular type of research article in study original lung cancer research. Then we're going to shift gears and talk about specifically reading scientific studies. So we're going to walk through that. How do you read? What are the strategies, the ways that you can get the most out of the article? You don't have to be a fancy scientist to understand and get the take-home message of the importance of the research. And then we're going to end with a resource toolkit. So 3 or 4 very helpful websites that I think can help you as you navigate reading papers now, but also in the future, and a couple tips and tricks that will help you understand how to look at these research studies. So we'll start with the introduction. So there are several major types of scientific articles, and you can think of them as 3 different flavors, all right? So we have primary articles, and these are original findings, okay? Then we have secondary articles. These are publications based on primary articles. So they're kind of like a summary. And then we have these special articles, so they can take various forms. So let's talk a little bit more about these different types of scientific articles that you would see the lung cancer research appear in. So there's original research, and that gives you new information based on studies that were done in a lab. And typically we see these are the things that are in headlines in the news. These are the breakthroughs. These are the things that come from these, fantastic clinical trials. A lot of people are talking about immunotherapy right now. That type of cutting edge, that is original research. But you also can have something called a case report or a case series. And that's when you have a description of a single case. So this could be one individual or individuals that have related cases, but there are some unique features. And that can tell you a lot about maybe treating a particular patient that has certain type of symptoms. And then there are technical notes. So this is a description of a specific technique or a procedure that may be used to diagnose lung cancer, for example. We talk a lot right now about low-dose CT, right? Modification of something that already exists, or even a new type of equipment. So all of this newness, right? Those are primary sources, primary type of articles. A review article, that's where there are these detailed analysis that summarizes the recent development. And they typically are narrowed down to a specific topic, right? And we highlight the key important points that have been already reported. So there are different types of review articles. These are very helpful sometimes if you want to kind of understand where the field is, a particular part of a lung cancer arena. You may want to go to a secondary article to prepare you to go back to read a primary article. And then there are these special articles. So for example, a commentary. Right? So this is a short article that describes typically the author's personal experience with lung cancer. This could be as a practitioner, it could be as a patient. There could be all types of perspectives in a commentary. An editorial, this is a short review or sometimes a critique of the original research articles. Again, that new finding that's been accepted for publication in the same issue of the journal. We also have letters to the editor. So this is a very short, objective, constructive comment, and this can be about any subject of interest in this lung cancer journal. And then finally, we have things that are pictorial essays. So these are more teaching articles, and they rely a lot on high quality images to help explain what's happening with the disease. So I just want to keep in mind that the primary articles, secondary articles, and these special articles, the one that we're going to talk about and focus on today, and is actually what most of you would see at a research conference, or maybe if you did a Google search, you want to look for primary, it would be the original research. So what does that look like? So just like our bodies have anatomy, research articles have anatomy. And similarly to when you learn more about anatomy and physiology, you understand how the body works. When you learn about the anatomy of a lung cancer research article, it helps you demystify and understand what's going on. Right? So we're going to take, for an example, one of my research articles. Okay, so this was a recent publication in Nature Communications back in 2019. So the first thing you're going to see is titles and authors. Right? So the thing about a title, you want your title to be very descriptive and also states the main findings. So when you're reading an article, sometimes you can be misled by the title. It doesn't necessarily match up with what's in the research article itself. And so that's one thing that'll tell you, hey, this may not be the most well-written original research article. So for the most part, the title should reflect what's in the body of this article. It should reflect the results and the findings. And then you're going to have a list of authors. So a lot of times people say, well, how do so many authors get onto a research paper? It's because they all have contributed something to this scientific story. Right? So just a little tip, when you see the author list, the order of the authors is important. Right? So the most common way that authors are listed on lung cancer research articles is by the magnitude of how much they contributed to the study. So the author who is first would have contributed the most to this research. So in this example, this is a first author paper because I spearheaded this particular study. Right? So next, you have an abstract. Right. So I like to think about the abstract as a movie trailer or a preview. Right? It's telling you what it is to come. So a lot of times, if the abstract is not well written, people will abandon that particular lung cancer research article because it's not clear if you should invest the time into delving further in that particular topic. So 4 things to keep in mind about an abstract, right, is that there's a brief background of the subject. So all abstracts function in this way. So I've underlined in red the brief background for this particular abstract for this paper. Right? It's putting into context the problem, in particular, how many people are getting lung cancer, how many people are surviving, and which groups in the country we're studying. Right? So next you see in blue here, the purpose for the study. And underlined in blue here, we talk about the purpose of this particular research study. So we were trying to look at DNA and find different biomarkers that could tell us how we could treat particular subgroups of patients with lung cancer. So then the main findings of the study. So these are outlined in green. Right. And so basically we found some new mutations that we were really excited about, and they are participating in a particular pathway inside some of the patients, and that let us leverage it for treatment options. So you see in green the main findings, and then in purple you see the relationship of these findings to the field. So basically, what does it mean, the findings? And you see here some of the keywords you see together these findings suggest. So you're saying, what is the point of this study? So an abstract is a really nice, concise summary to help you understand what's going on in that research article. So sometimes people will look at several abstracts to get a sense of what exactly is going on in the particular interest they have with lung cancer. All right, so how can you find these abstracts, right? So similar, you can go to IMDb if you want to look for movie trailers. So science, we have the same thing. If you go to PubMed, all right, or also PubMed Central, it's the sister site, and you type key search terms in there. So let's say for this example, I wanted to learn about lung cancer health disparities, and maybe I would also search for African Americans. If you put that in the search bar, you're going to get all the papers that have been published in lung cancer health disparities in African Americans will show up. And so the difference between searching in PubMed Central works the same way. You would type your search terms of interest and you'll get a list of abstracts for articles about your particular lung cancer topic. The only difference between PubMed and PubMed Central is the PubMed Central, you can get the full text of all of the articles for free. Some of the papers that are in PubMed are still behind a paywall. All right. So all the abstracts are free, but if you want to get the full text, the whole entire paper, you may want to start with PubMed Central first. Okay. So then we want to get actually to, what I think of as the meat of the article, right? So there is a very similar structure for all lung cancer research articles that are original in nature, and it's IMRD. Right? So there's an introduction, and what is the introduction section? That is asking what question about lung cancer is being queried here. All right. Then we have methods. So that methods will address how is that question studied using particular lung cancer research methods. Right. The results are going to tell you what new information was found out about lung cancer. And then finally, you're going to have a discussion. Okay, so in the discussion, this is where the authors are going to try to make meaning of the findings for a particular reason. So maybe they're going to talk about preventing, diagnosing, prognostic markers, so disease progression, treatment or management of the lung cancer. So this is a basic structure. Almost every journal uses this. Sometimes the methods may go behind the results or the discussions, but every one will have these basic components, the I, the M, the R, and the D. So let's walk through a little bit with the paper we're using as an example here and delve into a little more about the anatomy. So the introduction is typically 2 to 4 paragraphs, right? And it's going to give you all the background information to help you understand why the findings of this particular study are significant. So basically, what's the information you need to know to help you understand that particular paper and put it into context? All right. And so they have a typical structure. So there's a brief description about the current state of the field. So basically, what do we know about lung cancer right now in this particular time? And then there's a narrowed focus to a particular aspect of the field. So for our example, on the right, you can see the brief description of the current state is just talking about the statistics about lung cancer and the public health burden, and that it influences both males and females. Right? The narrowed down focus is talking about what particular segments of our population experience the burden of the lung cancer. So that's an example of what's happening on the right. And then there's the discussion of what knowledge gaps exist. So basically, what do we not know about this particular narrowed down topic? And so in this regard, African Americans are underrepresented in research and lung cancer research. So this paper was focused on filling that gap. And then there's a statement of hypothesis being tested by the current study. So basically, what is the new idea that's being pursued? And why would this research help fill in a knowledge gap? Right? So we talk about, I like to think about the introduction. If you think of a story, it's telling you what happened before and what's happening right now. And when we get to the end of the research article, we can think of what would the next chapter be. Right? So we're talking about what happened in the past in the introduction to get us to this moment in time to why we're doing this particular type of lung cancer research. Okay, so next we have the methods, right? So if you think about how you actually are doing these experiments to get the data, to get the result, to create this new knowledge, that happens in the methods section. So this can be a very daunting section. I remember the very first time I read a lung cancer research article, I thought, what the heck is happening here? I don't even know what this is, right? So that's a normal feeling to get in the methods section. I think the introduction, we kind of understand storytelling, but the methods is sometimes can be very technical, right? But method sections are written to the level of detail that another scientist can replicate that work. So basically, they can do the whole experiment over again. There needs to be that level of detail. But like I mentioned, this can be very dense and can require you to look up actually other articles to figure out what's going on. So just an example, when bold here on the right, we see this method section I'm showing from this paper. It talks about patient samples and DNA extraction. So it's talking about taking these lung cancer tumors and taking the DNA out of that. And there's enough detail there that someone can see who these patients are. They can also see the way that I took the DNA out of the tumors. They can go buy the same exact products. So a lot of times in methods, you'll see they tell you where you can buy those products. So as a reader, you don't need to always know those details, but it helps you understand how they did the experiment. All right. So then there's the results. I think many people are comfortable with understanding what a graph or a figure is because people are visual learners. Right? So the results, this we're going to see a lot of the visualization of a research article. Right? So some things to keep in mind about the anatomy of the results section. So it describes the outcome of experiments, right, that were done to answer the research question that was brought up in the introduction. And a lot of times the results are just stated. So it's just a sentence that says, this is higher than this level is lower than this. You don't interpret the results in that section. That actually comes in a discussion. So don't be surprised when you're reading a paper and it's like, okay, well, what does this mean? You just gave me this graph, because that is going to come later on in the research article, and that's standard and common. And thinking about figures and tables, That allows the reader to see the outcomes of the experiments for themselves without bias. So this is a great thing about the results section is that you can look at a piece of information and see if you agree with that or not. So on the right, I have an example graph here for discussion sakes. And we see that this is some new important lung cancer gene. And we're looking at two patient populations. We're looking at African Americans on the left in red and European Americans on the right in blue. And we're looking at the mutation frequency of this gene. So we would think that the higher the mutation frequency, perhaps it would be that more of those patients will respond to a particular targeted drug therapy that focuses on cells with the mutation. Right? And another thing that you will often see in results is, and this is a way you can tell if a paper, also an original lung cancer research article, is rigorous or not. So there's sometimes you'll see articles where people haven't put the appropriate rigor there. So that means, highest quality standard. So you should be able to see some type of statistics in your results. So these little stars here, that is indicative of a statistical test. All right. So you should see that when you read through the results sections, they'll tell you something may be a trend. That's a common thing to see, but it's not always that the trend is significant. So you want to be on the lookout for those type of things in the results section. And then there's the discussion. So when the results are analyzed, it reflects what the authors think are the key findings. Right? But it's really important to keep in mind that you as the reader, you don't have to always agree with the interpretation of what the authors of the original study said. A lot of times they see things from one perspective, and as the reader, you may not come to that same conclusion. So the discussion section talks a lot about what the authors think are the key findings, and it relates the current work to other findings in the field. So they'll often refer to, we had this finding and this result. This other group, who's on the other side of the world, had a very similar finding, right? So they're going to try to convince you as the audience that, hey, this is important, right? And we should continue doing this work to help advance the field, to help save more lives. And finally, the discussion section talks about how the current work is significant. So, for example, in this discussion section, we talked about there are particular mutations that were more prevalent in African American lung cancer patients. And because they were more likely to have a particular mutation, we actually did a lot of analyses and saw that they were predicted to respond to a particular therapy better than like their European American counterparts if they didn't have that mutation. So we're trying to put into context how you literally can change how treatment paradigms would work, right? Based off that research. So when you go to look at other original lung cancer research articles, the discussion section will be very similar. So they're going to take their findings and say, well, what does that mean and how can we use this to help in relation to what we know is also going on the research in other areas. All right. So the final two sections on anatomy are acknowledgements. So typically in that section, they are thanking individuals who contribute funding or research materials, or people who contributed to the research article, but not intellectually. So sometimes people provide technical support. Maybe it's submission of the manuscript and supporting data. It could be someone reviewed the article for grammar, so there would be an acknowledgement statement. So for this paper, the acknowledgement statement was to the National Cancer Institute because they funded my research study. And then finally, there are references. So many of us have written papers, high school, right, college, and so you have to have references, right? And so other studies that have been cited by this original research article are provided there. So there's an example of some of the references that came up with this paper. All right, so now we all, I hope, are comfortable talking about the major types of scientific articles, right? And thinking about the typical anatomy of an original lung cancer research article. So not an anatomy of a commentary or of a review, right, of those various, the special types of articles or the secondary. We're really talking about primary articles, right? New research. So let's go a little deeper into how you would go about reading one of these scientific studies. Right. So there are several approaches, and there are two common approaches that are used when you address a research project. So I'm going to give you these two approaches, and you could see which ones you think would work best for you. So there's the three-pass approach. Okay. So the three-pass approach means you're going to read this paper thrice. Right? The first pass, you're only going to spend about 5 to 10 minutes. So get your cup of coffee, you're going to speed read. You're going to skim. You just want to know the big picture. What exactly is going on here? Right? And then you're going to go and you're going to carefully read the title. Right? We know there's going to be a descriptive title. You're going to read the trailer for the paper. We know you're going to have the abstract. Right? You're going to read the beginning of the story, right? What happened the previous chapters. So you're going to go through the introduction and understand what is going to be new. But then you're going to jump to the discussion, right? And you're going to see what is that the authors are saying is so important about this work. So you're going to read that in its entirety. But you're going to ignore a lot of the nitty-gritty details at that point. So you're going to spend 5 to 10 minutes for a first pass. Typically, if an article doesn't pass the first pass filter, people will abandon ship and look for another article. So that is up to you. The second pass takes you about an hour. So you're going to read the entire paper to try to better understand what's happening. So now you're going to have to get a little bit more into the jargon, and you still are going to ignore some of the details, but not most of the details. So you're going to try to learn a little bit more about what is this particular method, right? Or what is this particular guideline that was given out before about screening lung cancer. So you're going to spend about an hour. And then there's the third pass. That can be anywhere from 1 hour to 5 hours. Right? You're going to read the entire paper for content understanding, and you're going to work to understand all of the details. And we'll talk about how you transition from first pass, second pass, and third pass. Depends on why you're reading the particular article that you're looking at. For many people, the first and the second pass is sufficient after you're comfortable learning to read these original research articles a little better. So at the end of the first pass, you're going to be able to answer these 5 C's. Okay, so you got category. That's the type of scientific article this is. And so you already know that. We just talked about these different types of articles. Context. What other lung cancer topics is this research study related to? Right? Third, correctness. Do the author's assumptions actually make sense? Does it really seem like this is the appropriate type of study to do to answer this question? Contributions. What is the study adding to the field? And clarity. Is it well written? So you can do all of that in 10 minutes, right? The first pass. For about an hour, you'll be able to, at the end of the second pass, grasp most of the article content. And you should be able to summarize the main points to someone else, right? And then finally, the third pass, you should be able to fully understand the article content. And not only that, but be able to criticize it, right? So you'll be able to pull out the strengths of the study, but also the limitations. So again, 3-pass approach. That's one great way, and these are the steps you would go to really go and understand a lung cancer research article in depth. But then there's also this 4-step approach. And in the 4-step approach, the first step, you skim the entire research article. So unlike the 3-pass approach, you're going to just take your time. You're going to look at the major headings. Right? You're going to look at how many figures there are and what type of figures are there. Maybe it's a microscope image. Maybe it's a graph. Maybe it's a table. So you're going to think through that, look at that, take stake in that. And then you're going to identify the take-home message. Even if you're not completely clear about all the details of what that means, you're going to be able to identify that. Step 2 is go through and master vocabulary. And I'll tell you how you can do that in a little bit later in the presentation. third, you're going to read for comprehension. So you're going to go section by section. Now, the interesting thing about this approach, you don't necessarily have to read a lung cancer research article the way you read a novel, right? Or an article in like New York Times, so to speak. You actually can start with different sections. Some people I know go straight to the results and they just try to figure out what's going on with the figures. They don't want to be biased by necessarily the perspective of the author. They just want to get straight to the facts. I know some people read the abstract first. There are people who skip the abstract and go straight to the introduction, right? So the point is you can read these sections differently and in different sequence, but you still need to read for comprehension. So whatever will help you. So you shouldn't feel compelled to have to read this in a linear fashion, so to speak. So step 4 is evaluating and criticizing the study. So you should be thinking, just like at the end of the 3-pass approach, there are questions and things you should be able to do after each pass. You should think the same thing here. So it's not necessarily with this approach, you don't have to do things once or twice or 3 times. You don't have to reread the paper, but you're taking your time going through and understanding each part. So at the end, you should be able to criticize this work and think about, were there enough experiments to actually support that key finding, that take-home message that we talked about in step 1? And were there some things that weren't there? A lot of times, maybe some of you are parents, you kind of know if you ask a question, well, what happened here? There's the things you see and the things you don't see, right? So in this regard, the same thing kind of like happens with lung cancer research. There's patterns and trends, and there's some things that aren't there that you may pick up and say, how come that wasn't in there, right? Do you agree with their conclusions from the data that you saw? Or is it a lot of hand waving? Is it overgeneralized, right? Sometimes you can see, like, for example, a headline about, there's a cure for lung cancer, and that may get us very excited, right? And then you go to read the actual article and you realize there's a lot of overgeneralizations. So it's important to be able to criticize the lung cancer research so that you can understand where it fits and how it can be helpful. And are there other reasons, right, that could have explained the results? And finally, do you have any questions? So as you're reading this, I think a lot of times people are fearful or like, oh, I don't know enough to question this or challenge. But a lot of times you can just take your intuition and your common sense and question what you're reading. And think about what would the next step be. So again, now I'm gonna give you some practical things to do as you read each one of these sections, the I, the M, the R, and the D. So how to read the introduction. So the first thing, I think this is what's different from when you read other types of lung cancer information, is that you actually should be taking notes when you read an introduction, right? And so here you're going to write down unknown words and vocabulary and jargon, or you can underline it. So sometimes people like to use a good old-fashioned notebook, but in this day and age, people have tablets. So there are all type of ways that you can take the PDF and annotate it actually digitally. But you want to take notes on this vocabulary. And then also, if you are visual, right, so people learn different ways, it's okay to know how you like to read, right? So you would want to draw many figures to help understand these relationships that come up in the introduction. So you want to ask yourself these 3 questions, right? What's the current lung cancer research problem being addressed in this article? And what is the hypothesis that they're trying to test? And how will this new research, how is that going to help address the problem? Right? So some tips for success as you're reading the introduction. You want to look up the definition in online textbooks. There's a ton of peer-reviewed free textbooks online now. You can go to Google Books, look at any type of lung cancer biology textbook, cancer biology textbook, dictionaries. I'm going to give you some of those in the toolkit, and also even online encyclopedias. There's genetic encyclopedias. There's all these things that you get information, particularly about lung cancer. And also take a break if you need to. So I think that similar as you read for leisure, when you're reading these original articles, it doesn't happen. You don't, just like you don't finish a book in a day, you don't necessarily finish sometimes a research article in a day. And there's nothing wrong with that, right? So you can pace yourself. The one thing that I would say is the benefit of taking notes and writing down is that you can go back and review where you were and just pick up. All right, how to read the methods. Again, this is the things, the nitty gritty things that you do. You want to take notes and write down unknown materials, things that you've never seen before in methodology. And you want to draw images in this section to help you understand how things work. So for example, I mentioned earlier about taking DNA out of lung tumors from different populations, right? So I will have a little diagram about how we do that, step 1, step 2, so that I can understand how did that DNA come out and how did you analyze that further. So the big question when you read a method section is, can you skim the methods enough to understand how the experiment was done, or do you need to know more? And if you need to know more, there are several things you can do to get more information. And this will demystify what these fancy technologies are. All right. So the first is quick reference images. So a picture speaks 1,000 words. Right. So you can go to Google Images and literally put a method in there. And a lot of times you're going to see beautiful images that explain to you how to do that particular scientific thing. Right. And how that's going to help how we use that to study lung cancer. And if you just want a quick reference, even Wikipedia often has images that show you how these methodologies work. If you're somebody like me, I'll be honest, most of my students, they go with the second option, videos, right? So if you go to YouTube, there every type of methodology video known to man is there. And so you can actually sit and watch a 5, 10, 15-minute video on how the actual experiments are done. So that actually puts it into life as you're reading these lung cancer research articles. You can actually see it performed in real time. And then there's something called Jove. A lot of people don't, if probably the public don't know what Jove is, but these are peer-reviewed scientific video journals. So if you go to jove.com, you can type your method in there. You'll actually even see scientists performing that particular experiment, and then that will help you get a better understanding of what's happening in the research article. And the last thing I want to leave you with about how you read a method section is something called citation chaining. So even now, I use citation chaining. So sometimes I'm reading lung cancer research articles, and it may be about touching on a field that I'm not familiar with. So I'm not a physicist. So sometimes there's articles that talks about lung cancer research and how physicists are using lasers to have very targeted therapy. And it's like, that's really cool. That'd be helpful. And so when I go to look at the paper, I don't know what's happening with the physics methods. And so we scientists who are experts in this are in the same place. So what do? We do something called citation chaining. And so you all can do citation chaining as well. And so what you do is, a citation is a reference, right? So what you do is you use search terms to go backwards in time, but also forward in time for articles that are cited by other resources. So in this example, the lung cancer paper that we've been using in this presentation, it has about 50 references. So if I wanted to learn about a particular method, I can go look in those references and go find the next paper that talked about how you do that method or how it was used in another context. And so you do this linking up, and then you'll have a chain of relevant literature about that particular method. So again, you can look at images, you can look at videos, and you can actually get other papers that explain how the method works. And then how to read the results. So we're talking about results and discussion typically together because the results are just a statement of what was found. The discussion tells you, but what does that mean? How do you interpret it? So you should be able, when you read a results section, to answer these questions. So with every figure or table that's there, can you explain to a family member or a friend the basic procedure that was used to make that figure or table? To question the figure or table was trying to answer, right? So do you realize, like, what was the point of generating this figure? What were the authors trying to communicate to the reader? And then the ways that the figure table could be improved. So a lot of times there are things that a figure or table can be confusing. right? And so what you can do is you can take a piece of paper and you can jot down an image that would fix it, right? That can help you better understand the paper. So strategies for success when you read the results section: read the text straight through, but as a figure or table is referred to in the actual verbiage, take as much time as you need to go look at the figure and to figure out what's going on. The figures are going to have captions, and the captions are going to say, this is, particular mutation changes, a bar graph reflecting mutation changes in these patient populations. So that's going to help you understand what's happening in the figure, and the labels and the colors and the keys and all those things. And the same thing with the table. So you shouldn't feel like you have to rush through a figure to understand exactly what is going on. And how to read the discussion. So you want to ask yourself, what conclusions do the authors draw? And can you separate the facts, which came from the results, right, from their opinion or how they're interpreting their data? And a strategy for success there is to describe for yourself why you think the results are significant, right? So I'll leave this section with giving you, there's a website there, it's really great. So Science Magazine is a common magazine that we use in the field, and it has a lot of different types of research, but really brilliant lung cancer research. But they have this commentary there that talks about, they ask scientists from around the world, how do you read your research articles? And so there are about 20 to 30 different scientists who are sharing their strategies for the way that they read research articles. And so that may be helpful to you all. And so I'm going to end here with talking about resources in a particular toolkit. So websites and some tips and tricks that can help you in addition to the strategies that I've provided so far. So first, it's thinking about helpful websites. So when we read these lung cancer articles, right, a lot of times there's terminologies and genes and things that we don't know, right? So we don't know what a particular gene is or a particular protein. We don't know, is there a treatment for this? What does this mean? And so there's a really handy dandy website called Gene Cards. And so Gene Cards, it's keyword searchable. And so you can put a gene or a protein or anything that's known to be related to lung cancer or may be described in the original research that you're studying. Maybe you never heard of that gene before, or you don't know what it is. So you can type that into Gene Cards, and it's going to give you user-friendly information. So it has every gene in the human genome, and that also includes the lung cancer genes. So humans have about 20,000 genes, and we know that 7 of those are involved with lung cancer. So Gene Cards will be a great place, and not only that, it links to more original research articles. If you go there, there's a link to PubMed as well as PubMed Central, so you can read more. There is the NCI's, the National Cancer Institute. They actually have these dictionaries, and I love these dictionaries. My students use them all the time. I use them when I talk to communities. And so there's a dictionary that has cancer terms, and this is very non-technical language, right? So there's terms and definitions, and so there's a lot of reviewers, and they add terms every month, so it's up to date. So you click that link, you can see either the link to the Dictionary of Cancer Terms, so there will be lung cancer terms there. There is also a dictionary of genetics terms, because so much is known about lung cancer genetics, and particularly how it's tied to different therapies and treatments. So if you have questions about that, you can type that into the genetics terms. That would help you. There's also a drug dictionary. And so this NCI drug dictionary, it will tell you what drugs have been FDA approved and those that are in current clinical trials and how that relates to, that may be important for if you're reading original lung cancer research paper that's talking about treatment or management of lung cancer. So those are 2 links, but to 4 resources that would be very helpful. And then also there is something called, the NCI also has this lung cancer patient version. And so when you go to this site, when you click here, you can look at the different subtypes of lung cancer, and it'll go into telling you about treatment, overview, the genetics of this, the symptoms, and all of that will help you as you're reading the original research articles to get the language and the jargon to put into context, but it's very digestible, right, for everyone. And then to leave you with a couple tips and tricks, I think things that I wish someone would have told me before I started not only reading but writing lung cancer research paper is that original research articles, you should read them when you're awake, right, and interested in reading. Don't force yourself to try to get through a complicated topic if you're not in that particular space. It's kind of like writing. So just like people use the Pomodoro method, they write for 15 minutes and then take a break. You can do the same thing with reading papers. So when you're ready in the time of day, some people read papers at a certain time of day, right? So if you are going to break up a paper and read it over several days, make sure you summarize before continuing on with the next day. Right? And note taking and highlighting can be done in print or digitally. So the image I'm showing here, many people print the paper out and write on it, and that helps. But they do the same thing with PDF versions. Third, imagine you are me. So you're standing up and you're teaching about science. And so you're teaching papers and figures. You're thinking about this in your own words. And you want to teach this so you're talking to your family or your friend or your provider, right? Or the person you're taking, the caretaker, if you're a caretaker, the individual you're taking care of, right? You want to teach them something. And so if you're able to put the figures from the paper and the tables in your own words, right, that shows you truly mastered and understood what was happening in the article. And the last thing I think is something that very few people emphasize when we have this discussion about reading a lung cancer study is that every scientist puts together the research article with the expectation and the hope that others would read this and build upon it, right? And to have discussion. So these research articles, these are scholarship as a conversation, right? So these are scientists talking with the public, scientists talking with patients, scientists talking with other scientists through their work. And so it is very common to reach out to the scientists that performed a study. So in fact, it's so common that it's a part of the actual study. So if you pull up a paper, you will see a corresponding author. And so just like when people often use snail mail when they correspond with one another through letter, we still correspond with the scientists. There's a corresponding author. So if you have a question about a particular study or there's something you don't understand, you definitely can email and reach out to the group that performed the work, in particular the corresponding author, which is also on that article, and gain more insight. And so I am done. I want to thank you all for the time and your patience, and that I will available for questions.
Yeah, go ahead. If you have questions, you can put them in the chat box. There's a little word bubble at the top. While we're waiting for any questions to come in, I have a question. I think that a lot of people are looking at lung cancer studies, trying to figure out, well, will this new drug help save my life or help me have a longer life? But there's lots of different terms like overall survival or progression-free survival, and it can be hard to know really what that all means. Can you take a second to speak to that?
Yes, that's a great question. So I spend a lot of time actually with my students thinking through that and helping them understand and tease it apart. So basically, there are two general types of survival when we're thinking about lung cancer, right? We think about individuals who are surviving or have low or high survival. We would like high survival, right? But some people don't survive and they succumb to their disease, right? That is what we think of as disease-free survival, right? We think about somebody, it's related to the actual lung cancer. The overall survival is not specifically related to the lung cancer. It could be related to something else. It could be a comorbidity. Maybe somebody had diabetes and also maybe somebody contracted an infectious disease and this complicated their disease course. So when we see that the real question when we have disease-specific survival data and overall is what was the ultimate factor that influenced if someone survived the cancer or not, right? And um, that is challenging, I will say, for someone to understand if they haven't had particular training like in that, right? But I think that I would focus on when I see studies, research articles, I don't want to say I ignore, but I don't put as much stock into the overall. I kind of look more at the disease specific or progression free, because I know they're particularly talking about the cancer itself and how it's influencing the patient.
Great. And then progression-free survival is like how long before your cancer grows after you've been given the treatment, right?
Right. So basically, we tend to look at 5 years, right? So we go to 5 years and we say, if you don't see your tumor come back in that 5 years, that's great, right? That there's chances are very low that you're going to get a recurrence, so a secondary tumor, right? And very low that you would still progress on and have worse disease course. What happens is there's more research. I just read an article yesterday. It was out of a group in Italy that said for lung cancer, we really should push it to 10 or 13 years if you can, right? They actually went to 13 years because you get a more complete picture of the disease progression. But yes, typically 5 years, we look at the 5-year mark to see, and that's a good indicator of what would happen to your disease moving forward.
Yeah, I think that's really helpful because a lot of these new drugs that are, published about, are published, the thing that they're very exciting about is that they keep people alive for longer. So not every drug is, going to be a miracle and save someone's life, but sometimes giving them more life, more time without their disease growing before they have to go on another treatment, I think is really important. And I think that sometimes gets missed, that sometimes, what's exciting is that the progression-free survival is 12 months or 8 months or something like that.
Right.
Yeah. Everyone, you can keep asking questions. This is recorded and we'll distribute it widely afterwards. But I have another question while we're waiting for any others to come in. And that's, can you help people spot poor methods? So I think that that's really confusing because I don't know a lot about methods. That's why I married my husband, because he knows methods and he can help me. So is there any way we can say, oh, this is not strong data?
That is a great point. I don't have an easy answer for that, actually, Carly. I'm a straight shooter. I tell it like this. People are very crafty at hiding their poor methods. I think that the one thing that helps is to do kind of like what I said about looking at the videos and understanding okay, what is this method typically used for, right? And then kind of looking at this study and saying, is that what they normally use that for, right? So it's kind of like methods change often, right, to get to the same question. So I like to think about air fryers. Everyone loves air fryers, and so everyone is using the air fryer to cook all types of things. So people cook burgers on the air fryer, they cook it on a grill, right? You could cook it in a frying pan in the house. But I think very few people would take a glass plate and put a burger there and let the sun hit it and wait 5 hours, right? I guess maybe in theory that could cook the burger, but doesn't really seem like the best ideal method, right? And all type of other things can happen during there. Animals can come do these things. How do we really know that cooked the burger, right? So I think that the only thing that can kind of help is you got to get familiar with the methods and see how do people typically use this approach. Right now, we're in a time where we have an explosion of technology, right? To sequence, when we think about sequencing lung cancer genes, and the way that I explained sequencing, if you thought about a street and you took some steps, and what if at every step you knew what type of piece of DNA was there, right? Step 1, there's one of these 4 bases. Step 1,000, one of these 4 bases. Well, way back when our methods, we could only figure out step 1 in 1,000. But now we literally know every step, every one of those 1,000. There's so much data and information. And so the thing is, what do we do with all of that? right? And the reality is that is a challenge when it comes to the technology is exploding so much and we have so many methods now and trying to like sift through that, right? So that's one thing to keep in mind, to not feel overwhelmed. If you can understand the basics as we evolve, this will help you keep things in context. So I would say like understanding like next generation sequencing methods as a whole will help you with so many of these papers right now. Because before we would treat people, okay, you had lung cancer, right? You didn't know what exactly, if you zoomed in, it's like magic school bus, what's happening inside the cell. But now we know that information and we treat accordingly. So I would say that in general, it would help to know just kind of like the phase of the type of the method, next generation, right? Like that.
Mm-hmm. Yeah, and I think that something that's helped me is to look at the N, which is the sample size,
Mm-hmm.
and realizing that, I guess studies that are stronger have a larger sample size, but in lung cancer, that's not always possible because you are digging down deep into the genome and, you're going to find less people with each layer that you go deeply. So I think that kind of just like taking everything as a grain of salt and looking at multiple studies. So it's not, you don't take one study as gospel. I think this is especially true, like I found with nutrition studies, where nutrition is a really hard thing to collect data on because people aren't always truthful about what they eat, and it's kind of hard to find out exactly what they've consumed and things like that. And so you might have a big study and people say that they and then you have headlines that say things like bagels cause lung cancer and stuff like that, but you actually have to like dig down and see what's going on. So to me, that looking at the sample size and then also like looking at, are they testing something that can easily be tested, like looking at someone's genome, which you can do in science, or are they asking someone for subjective thoughts or to reflect on how they behaved and things like that, which might make it a little more challenging.
I see a question.
Yeah,
actually.
okay.
But Carly, I want to respond to something quickly with that. So thinking about the method, when you mentioned the sample size and how many individuals are included in the study, there also, you may want to look for power analysis. So sometimes in the methods, there will be a statistic that'll say they did a particular power analysis. So they will tell you if their study is powered enough to actually find a significant finding, right? So so that's just something that I wanted to say there. Oh, ML, is there a way that their hand is raised?
Yeah, if you, I think it says Marino one, if you want to unmute yourself, you could ask your question now. Or you can type it in the chat box. Okay. Well, maybe it'll come through.
Okay.
Or you can email it to us and we'd be happy to get back to you.
Sure. I would like, oh, and I also see there are some comments. I believe the presentation.
Sorry.
Oh, there you go.
Oh, okay.
Hi, I'm sorry. I was unable to type in the chat box. I don't know why. I was trying to do that many times. I have a quick question. I have my mother. She's 82 years old, and she has a tumor, lung tumor in the right lung. Is healthy food very important to stop the tumor from spreading or expanding or growing?
That's a good question. I would say that obviously you have to talk with your provider because it depends on what type of tumor, right? One thing that we do know, Marino, is that when we look at diet, we know that there is some tumors are addicted to sugar, right? And this can worsen the progression. People have looked at prognosis, things like that. So of course, healthy diet helps us with your immune system being tip-top shape. So it helps you with managing like your condition. It helps you when you have treatment, Marino. So like if your mom has any type of treatment, we know that there are some components of the diet that can help those treatments, right? So it's like a synergistic effect. So a healthy diet can improve that. But I wouldn't say the diet alone is going to exacerbate or cure or fix the tumor. I wouldn't say that, but I would say that's definitely important.
And I can add, because we just had a registered dietitian do something for us, and she was really helpful in explaining that, you know, all cells need sugar to live. So if you starve your body of all sugar, you know, even healthy sugars, then you're starving all of your cells, not just the cancer cells, which I thought was a very helpful way to look at it. But, you know, there's healthier ways to eat. You know, refined sugar isn't as healthy for you as fresh fruits and vegetables. But then the other thing is that that she brought up that I thought was a really good point in terms of nutrition is that you want the person to be strong enough to withstand treatment and to have enough weight on their body and not be weak. And so sometimes if a person's not feeling well, you want them to eat whatever they can eat so that they can continue on their treatment. And so in that case, you know, you want, if that person can only really tolerate milkshakes because of digestive problems or nausea or mouth sores or something like that, then by all means give that person that milkshake so they can continue on their chemotherapy or radiation or something like that. So if you go to our website, lung.org, and you look for lung cancer nutrition, we have a lot about that might be helpful for you.
Great. And I see there's a question in the chat about it may be helpful to have another session to decipher graph results. And I think that also is very important, right? That's a topic in of itself about the different types of data that we commonly see in lung cancer studies and how to understand what's happening with that data, right? So that's a helpful suggestion. Thank you, CLS.
Oh, I see that now. Okay, cool. Yeah, and we can talk about maybe not only having the recording, but putting out the PDF. I can talk about to Dr. Mitchell and see if you're comfortable with that.
Happy to share. Yes, happy to share.
All right, well, thank you so much for joining. Thanks again to Dr. Mitchell. This was very helpful. We'll be in touch with the recording and the slides and more resources. We're always working on things. Feel free to contact us if you have any questions. Please stay safe, get vaccinated, and we'll talk with you soon. Bye-bye.
Okay, goodbye.